Thank you so much for the wonderful introduction, both for me and for the seminar series. This is really wonderful. And it's definitely my pleasure and my honor to attend this seminar and share one of my projects with you all. So I'd appreciate comments, suggestions, and discussions. I guess as introduced, I'll present a project called Modeling Subcellular Spatial Variation of Gene Expression. So in this project, I actually did this while I was a postdoc at UMich. We focusing on model the subcellular level spatial variation of gene expression. So we may be familiar with the spatial variant gene detection tasks. But here we are sort of still working on spatial variant gene detection, but the pattern we focused on is at subcellular level. For example, if a gene is highly expressed in the nucleus, but comparatively low expressed in the cytoplasmic area. So I guess that's the main idea about this project. I'll get started by introducing some background. I guess the audience here may more or less heard about that these spatial omics technologies like spatial transcriptomics and proteomics has been nominated as the Method of the Year by Nature Methods in 2020 and 2024. So these are quite popular and cool methods recognized by the scientific field. In this slide, I try to show a typical visualization of a spatial transcriptomics dataset. So on the right, we show a slice of mouse embryo tissue. So this is spatial transcriptomics data. So by name, we have the gene expression data. So we can do the cell type annotation. So after we get the cell types, something cool here, we have the spatial location. So we can actually visualize the cell type in the tissue in their original location. So we can see the different cell types are colored by different colors. And from this tissue, we can really see the tissue architecture from the annotated cell types. This technology is actually stereo-seq, which is a high-resolution spatial transcriptomics technology. So if we zoom in, we can see some subtle patterns of gene expression and also the cell type. So this is sort of a typical visualization of spatial transcriptomics. We have the cell type, single-cell expression, and we have their spatial information. So we focus a little bit more on the spatial transcriptomics technology here. There are increasing number of technologies, like the technology itself, it's advancing. So these technologies these are technologies for capture the spatial gene expression data. It can be roughly characterized into two categories: the imaging-based spatial transcriptomics. We may heard about MERFISH, SIGFISH Plus, Zynium a lot. And another category is sequencing-based spatial transcriptomics. So yeah, Visium, I guess, from 10x, this is a pretty popular one. There are Visium HD and the stereo-seq that we showed on the previous slides. So given the popularity of this technology, a lot of data collected, people like us are actively developing computational tools so that we can make the most of use of the data collected as well. So we can see if we check the publication, there's an exponentially increasing number of publications generated in recent several years, including published and unpublished work under different type of licenses and with software shared on GitHub and also, of course, Bioconductor. Those methods highlights a lot of different aspects of spatial transcriptomics data analysis. From this figure, I'm sorry, the texts are a little bit tiny, but we can see the spatial domain detection, deconvolution, cell annotation, integration. Those are all popular tasks where researchers have developed computational or statistical or deep learning methods to analyze the spatial transcriptomics data. One thing that is related to the topic to this project is if we have high resolution spatial transcriptomics data, we'd like to do the cell segmentation as well. And after that, we may have the opportunity to look at the subcellular level signals. So on this slide, I hope to show one of the high resolution spatial transcriptomics data that we have mentioned and of which data that this project will be focusing on. So this is a 10x human breast cancer data. By mentioning high resolution, we mean that this technology enables the precision gene expression at subcellular level. For example, in this visualization, we can say those white boundaries are the cell boundaries, and those kind of white shadows are the nucleus staining that highlights the nuclei. And those dots with different colors corresponding to the expression of different genes. So we may have an impression that, okay, from this visualization, we somehow can see, for example, in some cells like this one, it seems those red dots are more in the cytoplasmic area than overlapped on the nucleus. So from the data at this resolution, we feel that we can probably detect some subcellular level signals. And then we'll talk a little bit more about how the data looks like for the subcellular or high resolution spatial transcriptomics. So on the left-hand side, trying to show how the sequencing-based high-resolution spatial data looks like. So if you are familiar with them, this is something very similar. Like show in this cartoon, we have tissue slice. Here we have two cells. We have capture area, which are very tiny, so that we can have multiple areas inside the same cell. In this case, in each capture area, we have probes that can capture the RNAs. So the data format is similar to the WISM as well. We have one matrix for spot by gene for the gene expression, and we have another matrix for the spatial location of each spot. For the imaging-based, I believe most of the imaging-based technology are in nature high resolution. So we have those point clouds recorded. So we have the location of those points, and we also have the color or pseudo color corresponding to the gene ID. So we have a bunch of point clouds, and this format is actually equivalent to the two matrix format as well. If we want to slightly modify the data format, we can see it's basically we have the gene expression locations, but at each location we can only have one count, so this becomes binary. But here in the sequencing case, we have those counts of genes. Yeah. So this project, given it seems this data set has the potential, and also there are a lot of motivations for us to study the subcellular localization. For example, there are literature showing the mislocalization of mRNAs can be related to disease like mitochondrial mislocalization, and some type may be related to unexpected cell division that are related to cancer. Another thing is the compartmentalized translation. It's also related to the protein localization, which is directly associated with the cell function. And also, if you consider microenvironment, the cell and cells, they are communicating each other, and for the neighborhood cells, maybe they're communicating with using a particular type of molecule, and this is also something that we're interested in. And talk about the microenvironment, and we know those microenvironment in tissues can be related to treatment outcome. So these are sort of the motivations that we are interested in studying the subcellular localization of genes, of mRNAs. So here, those cartoons show some potential like patterns that we may expect. But the question here is, do we have methods that's readily available that for us to apply to such high resolution data to detect those subcellular patterns? So in this project, we developed a statistical method to detect the subcellular localization. It's for both detecting genes with subcellular localization pattern, and also it can provide the estimated pattern for that particular gene. There are a few key features. This method it's based on statistical model, the non-homogeneous Poisson process model. It's compatible with arbitrary number of cells, because in some cases, actually, most of the cases, we want to borrowing information across multiple cells because of the sparsity. It's compatible with diverse ST technologies because we said imaging, sequencing, those technologies, the data actually encode similar information. This method is also scalable, and applying this method to real data, we were able to detect interpretable mRNA characteristics. So this diagram roughly shows how the developed method works. So we have a data preprocessing step because we want to allow information borrowing of multiple cells to enhance the power. And then that's our model fitting step. After fitting our model, we will do likelihood ratio test to detect the genes with pattern and also calculate the estimated patterns based on Bayesian model averaging. So I'll begin introducing the data preprocessing step. So the step I'm showing here is based on a sequencing-based spatial transcriptomics technology. It's 6-scope. It's a UMIC technology. So for this particular data set, we have the H&E standing image on the same slide at the gene expression data. So we apply cell segmentation model. We use the cell pose to get the cell segmentations. For the gene expression, because this is a sequencing data set, we were able to do the alignments using the star solo and velocytal option. In this case, we can get the unspliced and spliced counts separately. So here in this figure, this is a figure from the 6-scope original paper actually. So we can see if we visualize the mitochondrial gene in blue, the spliced counts density in green and the unspliced counts in red. I believe we can see the cell structure here, like the intention is there's a cell nucleus and we can see those unspliced counts pretty enriched there. And this green intensity highlight the cytoplasmic area. We can see there's a hole, which is the nucleus, and the mitochondrial gene also show up in the cytoplasmic region. So this is kind of one of our motivating example where we can say the subcellular localization. So we got the unspliced data, hope to detect where the nuclear center is. So the first stage of the data preprocessing is really we get the cell segmentation and we align the H&E image with the gene expression so that we can transfer the cell segmentation boundary to the gene expression space. And then we detect the nuclear center, as we said, using the unspliced data, where we have in each cell, we define the location where we have the maximum intensity of the unspliced data as our nuclear center. And we do cell type annotation because when we want to borrow information across cells, we definitely want a homogeneous cell group. Here, cells from the same cell type can be an option. And we did some quality control to remove the cells with bad cell segmentation or genes with low quality. So that's the first stage data preprocessing. It includes modality alignment, etc. The goal of this step is really to obtain the nuclear center information and the cell segmentation information. So that's the outputs that we will use for the downstream steps from this stage. And then we move on to kind of the second stage of preprocessing, sort of an important step, because we said in many cases we want to borrow information across multiple cells, but these cells are with different shape. So we'd like to register cells into a common template. So suppose here it's some random cell. It looks like this. We're focusing on one gene and we look at this particular spot or transcript. We can calculate to distance. So we draw a line between the nuclear center detected in the previous stage and this transcript and extend it to intersect with the cell boundary. So we can compute this D1, we can get this D1 distance and this D2 distance. So we will calculate a relative position by D1 divided by the sum of D1 and D2. You may know that this is essentially a ratio. And this ratio measures how far away this spot to the nuclear center. So in this way, we actually registered a cell with random shape to a unit disk because everything becomes between 0 and 1. Yes, that's this step. And we can show a few visualization to check this. Actually, this type of registration makes sense because it roughly preserves the localization of the transcripts. Like if in a cell, those transcripts are in the cytoplasmic area, after we register them into this unit disk, it's still kind of reasonably in the kind of expected area. So that's this step, which is for us to borrow information across multiple cells. So that's sort of a summary of the data preprocessing step. We got some cells and we register them into unit disk. And the output of this step, it's actually we only keep the one-dimensional information. So we don't look at the angular position of those registered dots. We only keep how far it's from the nuclear center. So we are able to put all of these spots on a axis, one-dimensional axis from 0 to 1. So 0 means exactly nuclear center, 1 means exactly the cell boundary. And we can see we have those transcripts or spots registered here. And these are Rij for cell I and gen J. It measures the registered, the relative position, how far it's to the nuclear center as a ratio between 0 to 1. And this spot also comes a count. Like for the sequencing-based method, this is a count, but for imaging-based method, so this is only this can only be 1 because it's either observed or not observed. So this is the output of the preprocessing step. And then we will propose our model to the preprocessed data on the one-dimensional axis. So we propose a non-homogeneous Poisson process. The model is basically this index J is for gene for gene. So we are looking at one gene at a time. So for this particular gene, we have multiple cells that we want to pull together. Our idea is if we look at an interval within within 0 and 1, so if we look at a particular interval, we sum the counts within this interval together, and those counts should follow a Poisson distribution with this intensity. And this intensity is an integration from A to B, the interval, and some intensity function. So this function is on 0 to 1. So you can imagine if I choose different interval A and B, this integration going to be the same. So it's going to be, it's going to be different. So it will be different Poisson distribution for different interval A and B. So in this way, we model the variation along this 0 to 1 interval by this lambda star intensity function. So we actually further decompose this lambda star function into 3 terms. This Ci, it's only related to cell I. So we use it, we use the total count here, because it's some cell level feature. We have this SR, which is for normal, it's a normalizing term for the area because we only look at one-dimensional, but actually the cells, it's in 2-dimensional space. So these two are kind of things, covariates we want to adjust. And after adjust these two, we have this lambda R, which is the function shared across multiple cells for this gen J. So this lambda R, it's the expression pattern that we want. We have some slightly different version of the model where we add a random effect here just to further, because Poisson, it assumes equal expectation and variance. So just to release the model assumption a little bit if needed. So we said our focus is really this lambda R parameter because it estimates the pattern of this gene across by pulling information across cells. So to estimate this function, we actually used kernel fitting method. So we proposed several pre-specified kernel functions. Those functions are defined by beta PDF, because beta PDF by nature, it's on the support 0 to 1. It's a PDF, so the integration equals to 1, which makes our computation easier. So with these showing in the visualization, those prefixed pre-specified kernel functions, we feed those kernel functions one at a time. So for each kernel function, we allow a linear transformation. Those alpha and beta are unknown parameters, which will be estimated by fitting the model to the observed data. So by fitting the model, we are able to get the MLE estimate using the ADAM algorithm. So we will have this estimated alpha and beta, and thus this estimated lambdas from each kernel. So we are interested in detecting if a particular gene has spatial pattern or not. So in this case, it's kind of simple because it's equivalent to testing if beta is 0 or not. Because if beta is 0, then our lambda, given the model specification, it equals to this alpha, that's become a constant, then corresponding to this random pattern. But if this beta not equals to 0, it means our intensity function has some projected pattern corresponding to this kernel function, then it may have some function. So we'll do hypothesis testing, likelihood ratio test, testing if beta equals to 0 or not. Then we will have one p-value for each kernel function. So we will use Cauchy combination to combine the p-values across kernel functions so that we can have one single p-value for one gene. In the case if we have multiple genes, we'll need to do FDR later just for the multiple testing. So that's the testing, how the testing looks like. And we also want to have the estimated pattern as well. But by now we have the estimated pattern per kernel. We definitely want to integrate the functions across the estimated pattern across kernels, because each kernel can only capture one aspect of the unknown pattern. So we'll do Bayesian averaging to average the estimated lambda from each kernel to one overall estimated pattern to be our pattern. So I guess a summary of the outputs of the proposed method here is for each gene, we will have an p-value from the Cauchy combination, and we will have one estimated pattern from the Bayesian model averaging. And here are some cartoons. So we're expected to detect, for example, if this red transcripts red gene is pretty nucleus enriched, we will expect the intensity function to be high, close to 0, because 0 corresponding to nucleus center, and low as it goes to 1, corresponding to the visualization here. So that's sort of the idea of the method. So after proposing the method, we did some simulation to compare with existing method and also to evaluate the proposed one. Yeah, there are these competing methods, they're pretty good methods as well. And we are trying to show here that our method is pretty widely applied across different scenarios, and also it requires the inputs that are usually available from those high resolution spatial transcriptomics dataset. So we first did some null simulation where we generate the gene expression that are randomly localized in some real cells, like the cell shapes are from real data, but the gene expression are generated using simulation. And we are able to see that our method, including most of the existing method, we have well-calibrated p-values under the null case. And here we did some alternative simulation using multiple cells, because we said in the real data analysis, we want to apply the method to multiple cells to boost the power. So we can see Ella achieved high power in those so-called symmetric patterns, because we generated patterns that are enriched in the nucleus or enriched in the membrane. So these symmetric patterns. And also we tested some asymmetric patterns, for example, the radial pattern, polarized pattern, or punctate pattern. And we found Ella performed pretty good in except for one radial uniform pattern. That's because Ella it's a one-dimensional method. If there's no pattern along this particular radial dimension, then Ella cannot detect it. So that's the alternative simulation across multiple cells. And here are the estimated expression intensity, and the dark blue dashed line are the true pattern. The light green lines are the estimated pattern. The green lines are corresponding to the not significant estimates. So we can see the green ones are pretty accurate. We also applied ALA to the single cell case because we can apply ALA to multiple cells. We can allow it to apply it to one cell at a time as well. So in this case, we compare it to bento. However, if we only have one cell, we do require the gene expression to be a little bit dense. So in this simulation setting, we consider 3 counts per cell, and we can see Ella achieves pretty good power across different patterns from nucleus to the boundary enriched patterns. And here's comes the real data analysis. We applied to several, and here we'll show 2 real data analysis results. This one is a sequencing-based data. It's the stereo-seq mouse embryo data that we showed in the introduction. So we focus on an area in the slice that is close to the embryonic heart, and we're focusing on 2 cell types, the myoblasts and cardiomyocytes. So we have roughly 500 or 600 cells, and we apply ALA to almost nearly 2,000 genes, and we apply the ALA to 1 gene at a time. The cell segmentation was obtained from the nucleic acid staining image using cell pulse, and the nuclear centers were identified based on the unspliced expression density as we introduced before. And here are the estimated patterns from Ella. Those patterns are clustered into different pattern clusters and highlighted with different colors. And we can show some demo genes from different clusters. For example, this red gene, it's pretty high intensity at 0. And we viralized 5 cells, overlaid them together with nuclear center at the anchor point. And we can say the viralization, it's consistent with the estimated pattern. And this viralization also sort of highlights why we need to borrow information across cells, because imagine if you don't overlay those cells together, it's very hard to say the pattern given this gene expression is so sparse, because here we have 5 cells and we can see there are only 5 counts here. And we try to do some validation to check if the subcellular pattern detected by Ella are make sense or not. So we check the unspliced and spliced ratio. For the nucleus-enriched genes, we expect the unspliced and spliced ratio to be high, because if we have more genes in the nucleus, and in the nucleus we know they are more likely to be high spliced isoform of the mRNA. So it makes sense for the unspliced and spliced ratio to be significantly higher for the nucleus genes, this red subgroup. We also checked the gene length, and we found the nucleus genes has longer gene length. One interpretation that from the literature, it's for the longer genes, it may take longer time for them to be transcribed. So maybe the cells are transcribed. Some would like to transcribes them stored in the nucleus and use them when needed. We find the transcription factors are more likely to be enriched in nucleus, and we find the ribosomal protein genes are more likely to be enriched in the cytoplasmic. So these are sort of validations. And we'll also mention another real data application here. It's a 6H plus cell line data. So this data has quite good throughput. So we apply Ella to nearly 3,000 genes one at a time in 170 each cells. And here are the estimated results and patterns, and we try to pick some demos, and we can see this data set has pretty good throughput, and we can see the gene expression pattern using one single cell instead of overlay multiple cells together. So we did some similar validation. So for gene lengths, again, we say that for nucleus genes, the genes tends to be longer, and for the transcription factors, the nucleus genes tends to has more transcription factors. So we got consistent results across platforms and across cell types. So we mentioned that this data set has pretty good throughput. So we know that cells from the same cell type can be a homogeneous group, but we know the subcellular pattern can be affected by cell cycle as well. So in this data set, we further clustering the cells into different cell cycle subclusters, and we apply ELA to the cells in each cell cycle subcluster as well. So in this figure, we try to show for the cluster 1 genes, so we identify them to be a nuclear cluster 1 when we apply ELA to all the cells. So now we apply ELA to the subgroup of cells belongs to each cell cycle. So here are 3 subgroup of cells. We try to say if there are any pattern. So if we pick cluster 2, we can see we were able to identify a subgroup of genes where their subcellular localization pattern varies across the cell cycle subgroups. So here we visualize the pattern score, which is the peak location of the enrichment, like at which position this gene is highly enriched, for example, at nucleus or at membrane. And we can say this peak location can vary across the cell cycle phases. So which kind of reflect the dynamics of subcellular localization across cell cycle phases. So that's the second real data analysis. ELA comes with the software. I know we are almost time, so I guess I'll skip this part. So here's a summary. Yeah, we develop a statistical method for detecting patterns, either one cell or multiple cells if you want to borrow information. Yeah, it's can be applied to different platforms. I think that's all of the things that I hope to share, and I definitely hope to acknowledge my lab at UMich because this is where I did this project. Also, I want to thank my current institution, Texas A&M, and also Central Children's Research Hospital. And also, thank you so much for this invitation, and thank all the audiences.